All right, so thanks, Hal. Thanks to the Sloan Foundation to make this happen. It's been a really nice session, I think, at least until this point. I'm going to pick up the ball that Anne dropped this morning with development or providing guidance on how to use qPCR in practical for practitioners in practical situations, and tell a little bit about the work that we've been doing in a way to make this method now available to practitioners, really to go into buildings and measure microbial contamination also with quantitative PCR. So as Anne has been mentioning, we have this decree for housing health coming now into force, and we have an application directive on what actually how to implement this decree. And what we're doing there is now we're trying to, next to cultivation, also provide guidance on quantitative PCR as a method in indoor microbial assessments. So I will not go very much into detail about the qPCR method. I had a few slides there, but I thought it's most of you probably know about the principle. But in essence, you still gotta collect your sample. It might be a surface sample, a building material sample, it might be an air sample. You need to process it. And for example, what we are doing with the building materials is that we use the suspension that could also be used for cultivation and throw this into the qPCR. You need to extract DNA, go for your qPCR reaction, which really is a selective amplification of the target DNA in your sample. What we've heard already today, you see what you're looking at, and finally you got a calculation, interpretation of results. So I've seen on a slide today, I don't remember what it was. It was speed. So what's the main advantage of quantitative PCR or of molecular methods in general? It's speed. It's one. I'm not sure is it the main advantage. First point, probably we could have a discussion like half day about this, whether that's an advantage or a disadvantage. At least I think we are happy that we are not restricted to only see what grows on our agar media, but that this method is not restricted only to culturable organisms. But we also see non-alive microbes, so dead material matter that accumulates on surfaces, and I think that's a discussion. Do we want to see only active growth or everything that is there? In terms of health, we know that also dead cells matter, so that's one argument. The processes are largely automated, and if you think about once you have established the method in your lab, experience of the lab person is not the key factor, which is very different from cultivation, where you really need somebody who knows how to interpret what she or he is seeing on a plate. The methodology is basically available. There's plenty of qPCRs around, and USA EPA has done a big job with that. And the charge cost for analysis per sample, I think, well, I'm actually not quite sure, but I think it's at this point comparable to cultivation, depending on how many assays you're going to run. But I've been looking on a few web pages, and it seems to be like that. So in terms of limitations, setting up the whole thing really takes, like, you need to pass a certain threshold. It's cost intensive, not only because of the machine, but you need different rooms where you do your sample processing and DNA extraction. Okay, I'm not going to move. DNA extraction, and then also the PCR. You're not going to mix this up, so you need space. And it's laborious to get everything up and running. Once it's running, it's straightforward. It's a targeted methodology, once again, so you see what you look for. The beauty of cultivation is, whatever grows on your cultivation medium, you'll be able to take into consideration in your interpretation of the result. Whereas in qPCR, if you look at Cladosporium genus, you're going to see Cladosporium genus. If you look at a larger taxonomic group, you're going to see what you're really targeting and nothing else. Limitations, that's something that I will go in a little bit later. You have to be worried about inhibition in your PCR reaction, which you can overcome. Especially, I think, for building materials, that's an issue. And the quantification is not always accurate. So if you do qPCR from a species with a species-specific assay, you're going to be pretty fine. If you want to do qPCR from a larger taxonomic group, so you're including Penicillium aspergillus species into one assay, you got to do certain compromises, because you're not going to get every single species being amplified at the same quantity. So that's a limitation. All right, so what we did with the with the building material samples in trying to come up with some guidance is essentially we try. So we set up a database. Mark told the other data about database is a magic word, I guess, whenever we want to now start applying new methodologies into practice. So we looked at qPCR determination side by side with cultivation from a total of some over 900 building material samples. And our idea was to suggest a guideline value for categorization of a sample into probably damaged or non-damaged. So we did the cultivation, traditional methodology, qPCR determination. We did a bunch of assays, burned down to a handful of assays that we thought are interesting in our approach. There are other approaches out there, and laboratories also use other things, but that's what we came up with. So what we see, and that's not really big news, well, qPCR levels are times higher. That's a big surprise. If you, for example, compare cultivation-based total functional counts versus qPCR-based total functional counts. And that's a Finnish lesson, by the way, so I'm sorry about the slide. So that's a cumulative distribution of sample, percentage of sample on the y-axis versus levels. So what we're seeing here is like the median, basically. So if we see that there's about 2,000 CFUs per gram is the median of the samples in cultivation-based, and we have about 200,000 in PCR-based. There are things we know, and the methods correlate moderately. They correlate significantly, but moderately. Well, we are measuring different things in these two methods. So how we try to come up with I'm laughing because I just had a backflash of I think probably 20 meetings where in the beginning of the meeting we had to remind ourselves what's sensitivity and what's specificity if we compare two methods. And I'll try to get it right now. If we think about sensitivity and we use cultivation as the gold standard method, then it would mean that a high sensitivity would mean whatever is positive, meaning damaged in the cultivation approach, you always categorize also positive or damaged with the qPCR. Specificity would mean that if it's negative in cultivation or non-damaged, you also want to be non-damaged in cultivation. And one thing that so what we did, we used our 900 building material samples and we categorized them based on cultivation threshold above 10,000 CFUs per gram, and we can have a discussion about that. We also included in the definitions some indicator organisms to make this more specific. Didn't make a big change. So anyway, we split the data sets into damaged, non-damaged, did qPCR assays and tried to come up with a cutoff that looked about right. But what you realize is that if you try to increase sensitivity, so meaning you lower the threshold in your qPCR so that you really get everything that is damaged in based on the cultivation, you also detect in qPCR, then you drop your specificity. So you get a lot of false positive results, and well, false positive is also another thing to discuss. So basically, we decided one thing we did is we said, okay, we need to combine more than one qPCR. We need to combine several assays that we get a better estimate. And the other thing is that we said, okay, we make it more important to have high sensitivity. So if cultivation detects, we want to be there. And the way this turned out, so we came up with a couple of assays that we combined. One would amplify Penicillium aspergillus fungi, it's a bigger taxonomic group. The other one does total fungi, so universal function, whatever you want to call it, and it's a lot of pitfalls, but it seemed to work quite nicely. And the criterion that we're probably going to suggest is, if you have in your building material sample either PENASP exceeding 40,000 cell equivalents per gram or total fungi exceeding 500,000, then your sample is likely to be damaged. And that gives us a sensitivity of 90%, so meaning 10% of samples show up in cultivation, but we don't get them with qPCR for different reasons. Specificity, meaning of 64%, means like we have 36% of samples that are non-damaged based on cultivation, but are damaged based on our qPCR criterion. And in this 36%, I'd not like to talk about false positive because, of course, we do 5 minutes. We do detect dead cells. We do detect what we cannot culture. So there's a lot of, it could be right positives. Okay, so I'll just jump over this. We did a bit of validation in terms of looking at repeated DNA extractions from our building material samples and see how well is the correlation, and we were actually quite happy with it. Of course, sometimes you get a sample, and these are the 5 different repeats of each qPCR, not of the qPCR, but the DNA extraction from the same sample. You know, if you're working close to the cutoff, then you might get, in your repeats once over, once down. So we might advise that, do duplicate extraction in parallel and so on. Okay, so to finish this off, and I'll just jump very quickly, we did similar thing with air samples, and it was so much better and so much easier because we didn't need to use cultivation to relate to, because, it's obviously arguable. Where we said that we have moisture damaged homes where we know from inspections there is a moisture problem, and we have reference homes, and so we collected air samples there in parallel, again with cultivation, Anderson impaction, like traditional methods, and then with button inhalable aerosol sampling for at least 60 minutes, so going from 1 to 2 hours about the air sampling, so which is already like an advantage in our experience over like a short-term 10-minute air sample. And what we see is basically, and now we're looking at indoor samples from damaged homes, indoor samples from reference homes, and outdoor samples. And this is now total cultivable fungi. We do see there is a difference. We can differentiate to some extent, and this is 25, 50, 75 percentile. So we can differentiate somewhat, but there is quite a bit of overlap. If we look this with, if we run total functional qPCR from these samples in parallel, we see a somewhat better separation, huge outdoor box there, like which makes sense, total fungi. You'll get a lot from outdoors. And with Penicillium aspergillus assay, something that we were most happy with, a much clearer separation in terms of how damaged versus reference homes are separated. I'm seeing already Mia moving a bit nervously. Of course, we don't make an interpretation in cultivation only based on total counts, but we do take into consideration indicator taxa. We have another value of 250 that we're going to come up with maybe. We looked this with a seasonal variation. That's interesting. We realized that we did a good job to advise with cultivation based to only sample in winter in Finland, where we have like snow cover, because there you actually can separate in index and reference homes. With the qPCR, it's in the same sample material. This looks much better. It seems that we can actually, throughout the season, separate the damage from non-damaged home to a big extent. Okay, last 3 minutes maybe I'll try to make it. What I'd like to talk about only that, of course, it's rather obvious that guideline values are really useless if we don't tell people how they should, how you got to do it so that you get there. There are many aspects, like in any method or in cultivation, especially if we compare it to where many aspects that really matter in terms of your result in the qPCR. Sample processing, it's actually, that's the same for any method. And DNA extraction, qPCR assay, there's a bunch of different assays out there that measure total fungi, and you can be pretty damn sure that whatever assay you use, you're going to get a different number. Standard curves is an important issue with qPCR assays, and I'm going to go there. So basically what you do in the qPCR, like you get an output from your qPCR measurement from the machine, which is a cycle threshold where the fluorescence exceeds certain threshold. And then you have a standard curve where you refer this cycle threshold to a known cell concentration. And I've been talking earlier about the Penicillium aspergillus assay. So if you do a standard curve, what we are doing is like we're trying to cover this range of different species with using 5 different strains, and they give you different amplification efficiency. So we try to be somewhere in the middle. But of course, you can also do it with less. But I'm going to show you in the next slide how this affects. Another thing is the use of internal standards. So what we're doing is that we add DNA, non-microbial DNA of known concentration into a sample before we start the DNA extraction, and then basically at the end correct for inhibition, for losses in DNA extraction, and so on. So what you're going to see here now is a graph, and I've taken like I just did a simulation, seeing what this looks like. So this is Penicillium aspergillus 87 building material samples, which are plotted here, and then cell equivalents per gram on the y-axis. I just sorted the samples based on their concentration. This is what it looks like if you have a standard curve with this full set of 5 different aspergillus fungi with our normal setup. If you choose to be a bit more lazy and just do a standard curve with one or the other strain, you might end up either being, quite clearly below or quite clearly above, depending on which strain you pick. So it really matters to say, how did you do the standard curve? And finally, this is what happens if you don't consider the internal standard. These big stalactites, or whatever they're called, I guess, down there. These are samples where you have inhibition in your sample, and you think when you see the raw output of your qPCR, it's a low level, but in fact, from the internal standard, you see it's been inhibited, you correct for it, and that makes your result. So to conclude all this, I think when and this sort of links to what we've been discussing about where this might have to go at a conference like this. So if we want to apply molecular methods, or like with any other method, I think the people who apply the method and who interpret the results, they need to know how they are doing and how to get there and what they're really doing in the determination, in order to be able to make a sound interpretation of a data into a result. Thank you.